We are next going to hear from Professor Robert Langer. Professor Langer is the David H. Koch Institute Professor at MIT, the highest honor that can be awarded to a faculty member. He is one of the world's most cited individuals and the highest cited engineer in history. Professor Langer is especially renowned for his work on advanced systems for drug delivery and the enormous impact that these have on human health. In his presentation, Professor Langer will discuss his work to date and how this has led to numerous new therapies for disease and vaccine delivery. His work on new approaches for engineering tissues may help not only in the treatment of disease, but may also enable the creation of organs and tissues on a chip, which could reduce the need for animal and human testing. So we now look very much forward to hearing from Professor Langer on future vaccines and therapeutics, approaches to deliver mRNA and other macromolecules. Thank you.
Thank you. Well, thank you so much. Thank you, Laurie, and thank you, Bengt, and thank all of you. What an amazing meeting, and I think every time Laurie and Bengt do this, it just gets more amazing. So I thought I'd try to follow what you just heard, which was great, and tell you a little bit about how, when I was about many of your ages, sort of what I tried to do and how I failed over and over again, but eventually how we got to where we are today. So my background's different than a lot of you. I'm actually a chemical engineer. I got my degree from MIT in 1974. And what happened around 1974 was there was this gas shortage, at least in the United States, pretty big gas shortage. You had to wait in line, at least in Boston, for 2 hours to get your tank filled up for a car. At any rate, the consequence of that was that if you were a chemical engineer, you got a lot of job offers. And so almost all my friends that year, they went to work for oil companies. I thought, well, I'll do what everybody else does. So I applied to oil companies. I actually got 20 job offers, 4 from Exxon alone. And I wasn't that good, but they just had so many openings. But when I went there, they would tell you, like, well, if you could just increase the yield by these tiny percentages, that would be terrific. It would be worth, tremendous amounts of money, and they'd pay you a lot. And I remember, flying back to Boston after those interviews thinking, I just didn't want to spend my life doing that. It made me think what I did want to do, and rather than just follow everybody else. And when I was actually at MIT, supposed to be doing my thesis, I got very involved in helping to start a school for disadvantaged high school students. And I got involved in creating new chemistry curricula and things like that. One day I saw an ad to be an assistant professor at City College of New York to do just that. So I wrote them a letter applying for a job, but they didn't write me back. But I liked that idea, so I found all the ads I could to do something like that. I found about 40 of them, and I wrote to all those places, and actually none of them wrote me back. So that wasn't going so well. So I thought, what other way could I use my chemical engineering background to help people? And I thought about medicine. So I wrote to a lot of hospitals and medical schools. They didn't write me back either. But then one day I remember walking into the lab, and one of the older scientists said, Bob, he said, there's a surgeon in Boston named Judah Folkman, and he said, sometimes he hires unusual people. He thought very highly of Dr. Folkman. I won't say what he thought about me. But let's see if I should, the slides. So Dr. Folkman had this idea, for those of you that followed his career. He said that if you could stop blood vessels, that might be a whole new way to think about treating cancer. And he actually said it was chemically mediated by a substance called TAF, or tumor angiogenesis factor. Now, it turns out that this was very, very controversial when he came out with this idea, and he was criticized for it a lot. I knew nothing about biology. I still don't know very much, but anyhow, I thought it was really great. So I and didn't pay anything like the oil companies, but I thought it was just really a way to do something good with science. So he hired me to actually prove he was right, but in so doing, isolate what would be the very first blood vessel or angiogenesis inhibitors. Probably the biggest problem we had with this was there was really, at that time, almost no way to study blood vessel growth. So we had to invent one. And the problem is there's always background blood vessels in different places, but there's a couple of places where there are not many or none at all, and that is the chick chorioallantoic membrane and then the cornea, say, of an animal, like a rabbit. So our idea was we would put a certain type of tumor in these, like a V2 carcinoma, which would cause blood vessels to grow. But then we needed this, like a tiny little system, like a nano or microparticle, that could deliver any of the molecules that I was isolating. And all of these molecules we thought were going to be angiogenesis inhibitors. They were pretty large molecules, like proteins or larger. And but nobody had been able, I mean, the field was almost non-existent, to create little particles that could deliver large molecules. There's only one company in the world, actually, in 1974, really working substantially on drug delivery systems, a company called Alza. And so Dr. Folkman was on their board, and he went out and he asked them, could they help us? They had Nobel laureates like Paul Flory on their board, but they all said, well, not really. If you have a large molecule, it's just not going to be able to pass through any of these solid materials. And so, you're not going to be able to do this. It's kind of like walking through a wall, is what they said. But if we were going to solve this angiogenesis problem, we had to solve the delivery problem. So I spent 2 years working in the laboratory, experimenting with many different techniques. I actually found over 200 different ways to get this to not work. But eventually, I was able to make these little microspheres or nanospheres. You see a picture of one and one cut in half. And in 1976, we published this paper in Nature showing for the first time you could release large molecules. It's also the very first paper where people were able to deliver, nucleic acids and so forth. Actually, even 3 years after it, we came out with a paper even using this kind of approach for little particles for vaccine delivery. At any rate, I want to go back to 3 months before this Nature paper came out. So I got asked to give a talk. This was 1976. I'd never given a big talk before. This was in Midland, Michigan, at a big conference on materials. So I practiced this talk for weeks, and finally the day came, I got up and I gave the talk for 20 minutes. And at the end, I actually thought I didn't do that badly. I didn't forget too much what I was going to say. I didn't stammer too much. So I thought when I was done, all these older scientists, being nice people, would want to encourage me, this young guy. But when I stepped off the podium, a whole bunch of them came up to me and they said, well, we don't believe anything you just said. And they said, in particular, the molecules, like you said before, were too big to pass through the solid materials, and also the solvents I was using, like ethanol or methylene chloride, they destroy whatever we put in. And actually, things went downhill from there, if that were possible. I tried to get research grants, and my first 9 were rejected. They said, well, an engineer doesn't know anything about biology or cancer, so we shouldn't fund him. Anyhow, so that wasn't so good. So then, I really liked being a postdoc, but a lot of my friends said, Pauly, you shouldn't spend your entire life doing that. So I applied for faculty positions, not just because of that, but I applied for them, and since I'm a chemical engineer, I applied to chemical engineering departments, and no chemical engineering department actually would hire me. They all said, well, this bio ideas that they make no sense. You should be doing energy or oil or, something like that. So I didn't get any jobs. Finally, I got a job in the nutrition department. The reason I got it was the guy who was head of it, Nevin Scrimshaw. He was a very famous nutritionist, but he was kind of a benevolent dictator kind of department head. So he liked me, so he offered me a job. But he didn't ask anybody else in the department what they thought, which might have been okay, except for one thing, which is the year after I joined the department, he left. So a lot of the senior faculty decided to give me advice, and their advice is I should leave too. In fact, Mike Marletta, who's a very good friend of mine, he was on the MIT faculty then too, and he was giving a speech a few years ago when I won an award, And I'll just read to you what he said. He said, well, one evening I went to a faculty dinner. This is 1979 he was referring back to. I went to a faculty dinner at a Chinese restaurant with myself and some senior MIT professors. He said, a senior scientist sat quizzing us while smoking a cigar. He said, when the older scientist heard my concepts for drug delivery, he blew a cloud of smoke in my face and said, you better start looking for another job. At any rate, I kept at it. Fortunately, I'm still at MIT, but now we had the bioassay, so we could test different things. So we did, and I probably had 100 different fractions we tested, and 99 of them didn't work. But one did, and this is kind of a gruesome slide to show you, but there's no other way to show blood vessel growth. And it was really quite striking. It was published this in Science. And what you see is that you have this sheet of blood vessels. And we probably did like 2,000 rabbit eyes. I was working with Mike Klein and Ken Falterman, who were surgeons. And we'd see this every time on the left, whereas on the right, you can see the blood vessels are lower, they're sparser, they avoid the tumor. So this was really exciting. It showed that such molecules could exist and would really provide a pathway for all future inhibitors to be isolated and studied. Nonetheless, a lot of you obviously are involved in medical things. Medicine is very slow. It took 28 years from this paper before the Fourier-reverse blood vessel inhibitor would get approved by FDA. And that was, used a number of our techniques, but it was really great work by Genentech and other places that would make that happen, and spending billions of dollars to do this. Starting in 2004, though, something remarkable happened. What you see is one after the next blood vessel inhibitor gets approved by FDA. Avastin, which is the first of these, is probably the number 2 or 3 best-selling biotech drug in history. And they're not only used for treating different kinds of cancer, they're really the only pharmacologic treatments for stopping blindness in the back of the eye, like macular degeneration or diabetic retinopathy, which affect millions of people as well. Anyhow, I want to go back to the 1970s again. One of the things that we did in the mid-70s is Dr. Folkman and I filed for a patent, which might seem more common today, but then Children's Hospital had no patents. I had no patents. Anyhow, we filed for it 5 years in a row. It got rejected. And I remember the officials at the hospital came to see me one day. They said, Bob, you are wasting so much money, on these patents. They're never going to get allowed. They said, just give up. But I don't like to give up. So I started to think, how could we get this patent allowed? Legally, of course. And science really wasn't working. But as I told you in the beginning, when we first were doing this, everyone said it was impossible, it couldn't work. I thought, well, maybe somebody wrote something like that down. So I did this, what's called science citation search, where you look back and see who wrote about your Nature paper. So I did this in 1982. That was a 76 paper. I found a lot of good stuff here, but here's one. And I had no idea this was even written. It's always interesting to see what people have to say about you. But anyhow, here's one from 5 of the top material scientists in the world at that time. And what they wrote is generally the agent to be released is a relatively small molecule with a molecular weight no larger than a few hundred. One wouldn't expect that macromolecules could be released. And then they went on to say that Folkman and myself have reported some surprising results that clearly demonstrate the opposite. So I like that word, surprising. And I showed that to the people at the hospital. They showed it to the examiner. And he said, gee, he had no idea. He said, I tell you what, I will allow this patent if Dr. Langer can get written affidavits, written statements from these 5 people that they really wrote that. It's a true story. And so I wrote them, and they were all nice enough to write me back that they really did write it. And we got this really broad patent. So this had gone on now for about 10 years. And I always, like a dreamer, I thought, gee, if I write papers and I do this stuff, maybe somebody will use something that I did. And I don't mean just citing it like in a journal, but I mean really using it to help people. And certainly that wasn't happening. But finally, after 10 years, I got a call from one multi-billion-dollar company and then another. And they said, gee, we read some of your papers. We heard you got this patent. Can you consult for us? And we'll give you, a consulting fee and a grant, which for a young professor, that was really good. But most exciting to me was they were really going to develop this. And what happened is I did, get a better car from the consulting fee. But I did get the grant, which was great. But they'd work on it for about 6 months to a year, and they just both gave up. So that was really discouraging to me. But then a couple of years later, Alex Klebanoff, one of my friends, said, Bob, we should just start our own company. So we got the patents back, we started a little company, and we kept doing it. And then we also got other patents, licensed them to other companies. And again, if I just show you some pictures, I won't go through all these, but it led to products that are really affecting hundreds of millions of people, all of which are encapsulated in different materials and so forth. I mean, one in the middle, which we worked on with Takeda, that's Lupron Depot. Those are little microspheres that can deliver a peptide to treat prostate cancer or endometriosis. You need to put them in these. If you don't, you can't swallow big molecules and get them absorbed or take them nasally. And if you inject them, they're destroyed almost instantaneously. So this enables that to happen. That they've really provided the whole, and also lots of drugs, if you give them systemically, they can be toxic. A great example of where some of the work we did affected that are what are called drug-eluting stents. If you try to stop, if hopefully none of you have them, but if somebody has heart disease, that's one of the most common treatments there is. But a lot of times if you put a regular stent in, it causes what's called restenosis. So now what they do is they coat them with these materials and put an anti-cancer drug like Taxol in them, and you get 1/1000th as much, right at the site. And so those are used by millions of people all over the world. And of course, this isn't even limited to just medicine. It's actually provided the whole basis for a lot of what goes in aquaculture, which shockingly to me is about $250 billion industry, and they're used to help, spawning for preserving rare fish and so forth. At any rate, I wish that even the early work we did was a cure-all for everything. It hardly was. One of the things I'd notice, and other people would notice, is you've made a microparticle, and you inject it in, especially in the bloodstream, they can aggregate and stick together when they're really tiny, like a nanoparticle. Also, macrophages tend to eat them. So we and others, like Vladimir Torchelin, came up with special coatings, like polyethylene glycol, which is actually in Miralax. If anybody takes it, it helps you go to the bathroom. It's FDA approved. And this was a paper we published in Science about 30 years ago. It's been cited thousands, I think, with its 13,000 times between this and the companion paper we wrote. But at any rate, they really do make nanoparticles. You can see that with atomic force microscopy or with quasi-elastic light scattering. But to just show you how it works, this is an in vitro experiment with macrophages, and we put an orange dye in the little nanoparticles. And if you don't have the PEG, if you look on the upper left, you see in an hour, it's eaten, all of these. But when you put different designs, which we've worked on with a PEG, you can see that there's a lot fewer dots, and sometimes none. And you can take this even further. Omid Farukhzad, when he was in our lab, put targeting molecules on so that you could even target it theoretically to specific places. And now some of these are being used to target the liver. One of the things I realized when I explain some of this to people is I don't feel like I do that good a job. Fortunately for me, must have been about 10, 15 years ago, Nova, it's a TV show in America on PBS. They came and did some filming, and then they made a video in this. They had a show called Making Things Smaller. And I liked their video about what we did, so I'm going to show it to you.
He starts with a nanoparticle of anti-cancer drug. That gets encased in a plastic that releases the drug over time. That, in turn, gets a special wrapping that disguises the package as a water molecule to fool the body's immune system. And last but not least, the address where it should be delivered, a key that will only fit the lock of cancer cells.
I should tell you that a lot of the clinicians I work with tell me it doesn't blow the cell up quite like that. But anyhow, you get the idea. And of course, the idea, and you can start using this, and people are, for delivering small molecules. But what's particularly exciting is offers the possibility to deliver large molecules like siRNA and messenger RNA. And yet, if you talk to people who are experts in gene therapy, like Phil Sharp or others, they tell you that the biggest issue in gene therapy is, I'm not necessarily agreeing with them, but certainly one of the biggest ones is delivery. And as Phil said, we need to solve the delivery problem. So we've been looking at that for a long time. One of the things we looked at was polymers and other lipids, and I'll go over each of those a little bit. With polymers, the problem is if you use a polymer and you kind of come up with a new polymer, it takes a long time to make. You have to make many steps where you have what's called a monomer. You block it with something so it doesn't participate in a reaction. Then you have to unblock it. You have to do purification. A lot of these could take 30 or 40 steps. Bob Grubbs, he won a Nobel Prize in chemistry a number of years ago. He's from Caltech, and he called me up one day and said, well, Dave Lynn is my top student. He'd like to come work with you. So Dave started working on, could we really make large libraries of polymers that, but we had to, but do them in very few steps. And what Dave started doing, this was published in JACS and Agnavandi Chemie, is come up with sort of one-step methods of synthesizing polymers, where he'd use a Michael reaction and do a conjugate addition of secondary amines to bisacrylates. And so the beauty of this is that you could use a lot of different materials. I'll show you this in a second, but you do a single step. You don't have byproducts, and you don't have to protect or deprotect. So rather than do what I normally would do, is spend 6 months making one material, maybe we could make a whole bunch fairly quickly. And that's what Dave and Dan Anderson, who was another postdoc, did. They took 94 of these aminomonomers at the top and 25 diacrylates at the bottom, and they made polymers out of each, combining them. So we had thousands of polymers. And they also developed high-throughput screening methods for testing them. And we found, again, this is over 20 years ago, but we found that 46 of these were as good as what was the current standard at the time, polyethylenamine. And here's just one picture where it was compared to lipofectamine. And this led to a lot of widely used reagents, which are still used today. The other big area is lipids. And lipid nanoparticles, if you look at them today, there's really 4 components: cholesterol, a PEG lipid, and that's based on what I mentioned to you before, a structural lipid, and then what's called an ionizable lipid. Now, the importance of that, basically a company called Vical, and then there were others, Wu and Wu, in the 1980s, they started using cationic materials, cationic lipids or cationic polymers, to complex the nucleic acid. And that worked, but it caused a couple problems. If it was cationic, then you might get complement activation or toxicity. And also, once it's taken up by the cell, it's hard to get out of the endosome. So actually, Frances Arnold, who spoke yesterday, she had a graduate student, Dan Pack, and he came to join us over 20 years ago. And he and Dave Putnam came up with this idea of what we call an ionizable cationic lipid, which means that it starts out, neutral, but then because of the PK, when it gets inside the cell and the endosome, it becomes charged and it gets it out of the endosome. But because it's also neutral at regular pH, it doesn't cause toxicity. And then also, I put another paper, Peter Cullis in Vancouver did some great work on this as well with lipids. At any rate, then the question is, what should your ionizable lipid be? And of course, at this point, probably, even though in 2000, maybe only, the liposome field had been around for a long time, maybe there's only 40 lipids used and studied. So Dan Anderson started using some of the techniques that he and Dave Lynn had worked on, and they made a lot of lipids. I'll just go through this quickly. But basically, you can just, again, use these conjugate addition approaches. But in terms of diversity, again, you can have large variations in the R group. You can change the tail length and also the number of tails, so you get a tremendous number of different. So again, we had thousands of lipids for the first time. And we worked with a company called Alnylam, I was on the founding scientific advisory board, and Akina Kink, one of our students, led a lot of the work there, which would ultimately lead to the development of Onpattro, the first siRNA approved drug by FDA. But anyhow, this just shows that you can get knockdown with these lipid nanoparticles and at different concentrations. And then we made them better. We took our first library, where we had some good ones, And then we used additional chemistry, epoxide chemistry. And what we did is we selected amines from our original library with bias towards good performing ones. And first we tested it in vitro. And you can see the nice thing is the one on the right is what we used before, but we improved it between 1 and 2 orders of magnitude, just again by improving the library. So the point is you can make these more and more potent. And of course, if you do that, you'll get an effect with, and you'll get much less toxicity too. And this just shows over a 10-year period how we, with working with Alnylam and Acuitas, how you could knock this down in terms of potency, by about 4 orders of magnitude. So we were able to make them more and more potent. And eventually, all this work would lead, this is Onpattro, which is a rare disease that people have. It's called ATTR amyloidosis. And by knocking down the gene, you can it changes people's lives. This got approved in 2018. So that leads me, all this, to setting the basis for what I wanted to go over at this point. So, we'd done all this work, and in 2010, Derek Rossi, who was a young professor at Harvard, came over to see me. So, as was mentioned yesterday, there's pioneering work in some of this goes back even to 1990 with Wolf's paper, where he started looking at mRNA as a possible therapeutic. But a key paper, which won the Nobel Prize, as was mentioned yesterday, was by Carrico and Weissman, who were able to modify messenger RNA and make it, much safer in the body. So, but they did this in 2005. They wrote a patent. They tried to start company, and they spent a couple years, and unfortunately, nobody would fund them. I guess people just didn't think this would ever lead to anything. Derek had read about their work, and he started using it to change the nature tried to do the same thing to make iPS cells that way. He tried to get funding from Third Rock and other places, and again, they said, well, it's never going to work. So he came over to see me, and I thought this sounded really good, and I felt like, to me, there were 2 pieces to this whole thing. One, of course, is the mRNA, but the other is the delivery system. If you can't protect it and deliver it to the patient, that's big issue why nobody would fund it, because people didn't think it would work. In fact, as was also mentioned yesterday, no large pharmaceutical company started doing anything on this until the year 2020, which was Pfizer. At any rate, I thought, based on what I just went over, the work we'd done over the last 34 years, maybe that could help. And we'd maybe really done 5 things. First, for the first time, show that you can deliver drugs like nucleic acids. 2, come up with coatings so that they won't get eaten and they won't aggregate. 3, come up with molecules that are, say, ionizable, or at least that concept, so that they won't cause toxicity and they'll be able to be more effective by getting out of the endosome. 4, advance this idea that really you can make a lot of lipids and maybe find one that will be pretty good. And 5th, which I didn't mention, Rohit Karnik, who came to our lab, actually developed microfluidic methods to make these, which is something that, at least to me as an engineer, seemed like it could someday be scalable. So with Derek, we talked to Noubar Afeyan, who was at Flagship, who put up some funding, and we also got Ken Chen, who was a cardiologist, to help us. So we started Moderna in 2010. And of course, the idea, which was just talked about in the last thing, is that you take DNA, makes RNA, makes protein. So by 2010, protein therapeutics had become enormously successful. There's so many of them, different antibodies and so forth. But it takes a long time to make them. It's expensive. You have to have VATs. If you're trying to make a vaccine like flu vaccine, you might have to start a year in advance and guess whether you're going to go after H1N1, H1N2, and you might guess wrong. That's one of the reasons why flu vaccines are maybe only 40% effective. But with messenger RNA, perhaps you wouldn't have to do any of that. You could just make it, put it in a nanoparticle, inject it into the patient. This was the vision. And then the body would do all the work. And not only that, you might be able to treat diseases that you could never treat before, like intracellular or membrane-bound proteins weren't being involved. So anyhow, we started Moderna, and believe me, I don't want to give myself very much credit. But if you didn't have a company, you could never do what happened. And so we were able to recruit some great people like Stéphane Bancel as CEO, Stephen Hoge as president, and we were very fortunate to get Jack Sostak, who spoke yesterday, as chairman of our scientific advisory board. Nonetheless, even with that, Moderna would get widely criticized by the press, by the news media, by stock analysts, by Nature, and so forth. But I always thought the people at Moderna were great, and a lot of them were my own students. And what would happen over time is, in spite of all that, by 2019, we had 13 products in human clinical trials, 8 of which were vaccines, early stage trials. And then, as you know, COVID hit. And I want to show you what the Moderna scientists were able to do. So if you look at this timeline, the Chinese scientists published the virus genetic sequence on January 11th. It took just 2 days for the scientists to design the vaccine, put it in the nanoparticles, send it to our clinical partner, NIH, for testing. And the first patients were dosed in March, 2 months. And when you do these trials, people may not, a lot of people probably do know what I'm going to say, but when you always start these clinical trials, you start with a phase 1 trial, which is a small population of patients looking for safety. So you try to find the highest safe dose. So we did that, and we had, actually I thought very good results within the first 2 months. It looked very safe, and we had antibody titers. So Moderna is a public company. You have to announce the results. So we did. I thought finally, and COVID, that's a crisis. You hope that finally, I thought people would cheer us on. So let me show you the headlines from our local paper when we announced the data on the front page. So this is the Boston Globe, my hometown paper. I guess they really like me. But basically, this is what their front page headline is. This is not how you do science, with my picture right underneath it. So that was the phase 1 trial. But you keep going, and you do a phase 2 trial. That's a small population of patients for efficacy, and then a phase 3 trial, which is a large population of patients for safety and efficacy, and it's double blind. So we had that data by November, and that data was 30,000 patients, 15,000 get treated, 15,000 controls. And I'm on the board of Moderna still. I was on it then as well from the beginning. So, they broke the code, and it was pretty amazing. I mean, basically, of the 15,000 who were treated, not a single person died, not a single person went to the hospital, and it was 94% effective. Now it's been used all over the world. I'll show you, this is just a picture of it. This is just an example of the efficacy from the CDC when you look at unvaccinated compared to vaccinated with a booster, and so forth. There's only in the U.S. 3 vaccines that were really used, and the 2 that were by far the most effective were the Moderna vaccine. And the other place I want to mention, which was also mentioned yesterday, is BioNTech, which is a German company, and they again did a terrific job. They also are using messenger RNA in nanoparticles. There have been work with J&J using adenoviral vectors, but that was removed from the market. There are a lot of things that have been published and in top journals like New England Journal of Medicine and others. I'll just show you a few, a couple of different things. This is a very recent paper out of JAMA from Brown, just showing, with over 6 million patients, showing this is extremely safe and effective. And the safety issue came up yesterday. I thought I'd just show a little bit. First, are vaccines safe? And I think John Marilyn yesterday, made some points that certainly I would agree with. That vaccines are very safe, but you still, no matter what you take, any medicine is going to lead to side effects. But here are just 4 different agencies saying that vaccines are the most effective public health tool ever. And there have been studies on looking at the safety of the Moderna vaccine and the Pfizer vaccine. And this is just a study comparing the adverse effects. And what it shows, and I think it's important, and it's exactly what John Marilyn said yesterday, is you do get side effects. But if you compare the mRNA vaccine side effects to the ones you get with flu, probably they're a little bit less overall with the mRNA vaccine. So that's a little bit about safety. And just to go over, again, some data, to just show this has not been an easy thing to do. Vaccines, there are 49 failed vaccines, 2 approved. This is for the US. Treatments, because there have been 209 failed treatments, 1 approved. Antivirals, all for this, 153 failed, 1 approved. And there have been places that have tried to do analyses of what happens. And again, there's over 3 million, it prevented 3 million deaths. And this is all under less than a 2-year period. They're talking about prevented over 18 million hospitalizations. And again, this is just US alone, and prevented almost 120,000 COVID infections, saving over $1 trillion. I think it's also, since I'm talking about was asked to talk about the future, part of the future is doing this for a lot of other things. So the idea here again, I think it opens the door for what are called personalized cancer vaccines. And so this is one that Moderna has done with Merck. If people have advanced melanoma, what they're doing is you can take up to 34 mutations that are known as neoepitopes present in a patient's cancer cells, and you can put that code into the mRNA vaccine exactly the way I just showed you. You put it in nanoparticles. And again, this is another double-blind trial. Keytruda is probably the best immunotherapy drug around. Half the patients get this, and half got Keytruda plus PCV. And yet at 2 years, the risk of recurrence or death was reduced by 44%. At 3 years, 49%. Metastasis, that was reduced by about 69%. And if one looks to the future, again, and this is, I'm just going to speak for what I see from Moderna, there's a lot going on. The way to judge what will happen, at least in my opinion, in the next 5 years is look at what's in clinical trials now, because it takes a long time from clinical trials to real products. But I'll go through this very quickly. But what you can see are all kinds of vaccines. Someday I think you'll see combinations of like COVID vaccine combined with flu, combined with RSV, just like the way now a little kid gets Prevnar. You put many vaccines in the same thing. And then there's a lot of other vaccines like cytomegalovirus, Epstein-Barr virus. In fact, some people think this could be a way to prevent mononucleosis, and that in turn may cause multiple sclerosis. Many other types of vaccines are listed here. And then there's therapies, relaxin, again, different ways of treating tumor, heart disease, and then rare diseases like propionic acidemia, MMA, glycogen storage disease, cystic fibrosis. I'd like to think that finally, that after all the criticism we got early on, now people start to look back at that 76 paper and feel like maybe it made a difference. I wanted to also talk another thing about the future. I had a visitor come to see me about 12 years ago. He was actually a very successful computer entrepreneur, and he and his wife, they wanted to help people in the developing world. I'll just show a picture. Yeah, so that's me on the left, and that's the software entrepreneur on the right. For those of you that don't recognize him, as Bill Gates. And he's really a visionary guy. He said to me, this was 2012, he said, Bob, can you come up with better treatments, better vaccines, and better nutrition, better ways in the developing world? In particular, he was concerned that people didn't come back in the developing world for a second vaccine, often a second pill. And we're working on all those, but I just thought I'd give one example. And that's the idea of a self-boosting vaccine. Could you just give it once? And again, this is something we're working on, but basically, and we published a new kind of, I should say, kind of a new printing approach in Science, and followed it up with a number of other papers. But the whole idea is, could you, when you look at this little gray rectangle or square in the middle, that could be like a polymer, and the red part, that could be the vaccine. And the idea is, can you have it deliver at time 0? That's pretty easy. But could you also think of these as little eggshells? Could they break like the green one at a month? break like the blue one at 2 months and so forth. So could you make a vaccine, could you put the vaccine in here and then just have these little shells break at specific times? And so what Anna Jakulenik and Kevin McHugh in our lab did is we took a FDA-approved material, polylactic glycolic acid. It's one we've worked with now for many years, and make this printing approach and make these tiny little systems. And the idea, if you look at the middle bottom, the blue is like a dye representing the vaccine, and then you're covering it with this little eggshell, which we could make break, again, depending on the polymer nature, at any point in time. And we did that. Here what you see, this is actually in vivo. You're taking lactic glycolic acid, which I said breaks up into water and CO2, but by changing the molecular weight, the ratio of those two units, adding a buffer like calcium carbonate, you can make these break at almost any time. So we made these, so this is like one shot, and yet you get 11 pulses. And someday what we're thinking about doing, again, funded by with the Gates Foundation, is you could also put these or other vaccines in what are called microneedle patches. And the idea of these microneedles is it's like a little Band-Aid, but you have these needles, and they go in not very deep, but deep enough so they'll be able to deliver something, but not so deep that they cause hardly any pain. And so Mark Prausnitz, who was one of my students, he's at Georgia Tech now, he's done a lot of work on this. And so we've actually made these, and the idea is that you could put if you go to the one on the right, that's the microneedle patch. Here it looks like, as I said, just like a Band-Aid on the left. And then the idea is hopefully someday you could ship these all over the world. To that end, BARDA also, again, before, all these things happened before COVID, but because people were thinking about this anyhow, BARDA, which is another funding agency in the United States, contacted Anna Jakulenik in our lab and said, could you ever make a 3D printer you could ship to remote areas in the United States or to Army bases or underdeveloped countries? So what we did, this was published in Nature Biotech last year, is we made this tiny little desktop 3D printer that could print those patches. And you just see pictures of it here. And you can sort of see the needles on the right. But as we showed, it actually works pretty well. You can, if you compare in this graph, the antibody titers, we're now looking at the receptor binding domain, and you compare the intramuscular injections, that's in the middle, to the microneedle patch on the right, you get pretty much the same results. In fact, maybe even a little bit better. And we've even come up with formulations that stabilize these so you can ship them and they're stable for 6 months, even at fairly high temperatures. So I thought I would just end the talk with one other thing, and that is another totally different thing about materials. One of the things that I noticed being a chemical engineer is I started to notice, how do materials find their way into medicine? And I thought naively, this goes back again to the 70s, that it must be older chemical engineers or chemists. But I found that was never true. Pretty much every material that got into medicine, if you look in the last century, was a surgeon was a clinician going to their house and finding some object that resembles the organ or tissue they want to fix. So like the artificial heart, that's a ladies' girl material because this was 1967, because a heart surgeon, Bill Pierce, said, well, that's got a good flex life. But the artificial heart runs into problems. Blood hits the surface of it, forms a clot, patient gets a stroke, and they might die. And by the way, this doesn't stop with hearts, like breast implants. One of those is a mattress stuffing because it's squishy. So a lot of what we've done, and I didn't really go over it that much today, is really think about rational design of new biomaterials. And that's led to a number of other things that we've done that have hopefully helped people. But I thought I would, in particular, go over another idea, which is that you could combine materials with cells, that was mentioned in Laurie's introduction, to make new tissues or organs. This is a little baby. He's dying of liver failure. Jay Vacanti, one of my friends, who's a liver transplant surgeon, treated him. And Jay and I came up with this idea. This was published in Science. It's been cited about 9,000 times. And the idea is you could take any cell type, dissociate them, and if you inject them at random, almost nothing happens. By the way, you could also do stem cells. We wrote this before people talked about them. But at any rate, and then you'd convert the stem cells into one of these. So the idea is, if you just inject these tissues, right away, the cells right away, not much happens. But if you put them on the right scaffold, and I'm just doing this at a super high level just to show you what might be possible, and in the right reactor conditions, that's the in vitro culture, literally you can make new tissues or organs. This is just a picture of some of the scaffolds that we've synthesized, like polylactic glycolic acid, which I mentioned before, but also cells can't stick to that very easily, so we've synthesized other materials like polylactic co-lysine. So through the NH2 group, you could link, say, if for the chemists, through a carbodiamide reaction, different amino acid sequences. So let me just show you really quickly one of the things we did. This is a scaffold with cells on it, and here is a patient where this was actually used on. So this is a little baby, 2 years old, very badly burned. What you can do is you can take this scaffold and you can, this is done by a company based on what we did, and these are cryopreserved, but you can put it, and you take neonatal skin fibroblast, and they can be frozen, but you can put it in the child at the time of the injury. But let's come back 3 weeks later, and now 6 months later, and it's pretty much healed. And not only can you make these, but you can also begin to think about making organs and tissues on a chip. This was also mentioned, I think, yesterday. And so here's another study we did. Gordana Vanyak and Milika Ritzik in our lab. They're both now professors at Columbia and Toronto, respectively now. But here, they actually made a heart on a chip, taking cardiomyocytes and put it on, and you get the characteristic structure. But what's really interesting is you can take 6 million heart cells, cardiomyocytes, and what they do, and I'm not shaking this or anything, but you can see it beating like a real heart. And you can actually take an EKG of it. And they even have a little company, Milika and Gordana, which does drug testing, and it's working with about 20 other companies. And so where this is now, if I look at all the things that we've been doing in our lab, as well as some of my students, there's a whole bunch of these that are in various stages, either organs on chips or being tested. They go from cartilage, spinal cord, GI tract, hearing. This bottom one's a brain on a chip. Blood vessels, which has actually been used already to treat patients in the Ukraine who have been wounded. Vocal cords, that's Julie Andrews, liver, heart, skin, pancreas. Let me stop. Thank you so much for listening.